My name is Anke Lohnsdorf. I'm a senior consultant physician at the Department of Dermatology at Heidelberg University Hospitals in Germany and a senior author on the research team that has recently studied infiltrating immune cells in cutaneous squamous cell carcinoma of organ transplant recipients compared to immunocompetent patients. Why did we do this study? Well, squamous cell carcinoma of the skin is the overall most frequent post-transplant malignancy. In organ transplant recipients, the skin cancer burden and also clinical outcome seems to directly correlate with the duration and intensity of iatrogenic immunosuppression. Further highlighting the critical role of the immune response in the development of squamous cell carcinoma, in immunocompetent patients, the magnitude of the local inflammatory response seems important for the spontaneous regression and clearance by therapeutic modulation of actinic keratosis and in situ squamous cell carcinoma. However, the impact of immunosuppression on skin homeostasis and local immune responses towards squamous cell carcinoma is still a topic of ongoing discussion and research efforts. Our aim in this study was to most accurately and comprehensively quantify and characterize immune cell subsets in both the neoplastic tissue and at defined regions of the tumor vicinity in squamous cell carcinoma of immunosuppressed organ transplant recipients compared to immunocompetent patients, and characterize the microanatomical distribution of these immune cell subsets. We set out to achieve this by accurately matched tumors and patient cohorts, analysis of whole tissue sections as opposed to the analysis of only fractions of the tissue, and the use of highly objective and standardized methodology. Here is how we did the study. We selected formalin-fixed paraffin-embedded squamous cell carcinomas from 20 kidney transplant recipients cared for at the dermato-oncological outpatient clinic for chronically immunosuppressed patients at the National Center for Tumor Diseases in Heidelberg, Germany. Those tumors were carefully matched with squamous cell carcinoma from immunocompetent patient based on clinical and histopathological features such as gender, age at squamous cell carcinoma diagnosis, tumor location, and tumor grading. Histopathological features of all tumor samples were established by a board-certified dermatopathologist in a blinded fashion, and 2 tumors from immunocompetent patients had to be excluded due to insufficient quality of the specimens. The basic clinical and tumor-related characteristics of the analyzed cohorts are shown here in Table 1. Serial tissue sections of 4 micrometers were prepared from these tissue blocks, and we then joined forces with the group of Professor Niels Grabe from the Hamamatsu Tissue Imaging and Analysis Center at BioQuant Heidelberg in Germany, where these tissue sections underwent automated immunohistochemical staining for CD4, CD8, CD20, and CD68. Consecutively, whole tissue sections were scanned, digitalized, and semi-automatically analyzed. We selected the tumor margins on hematoxylin stained sections, and based on this tumor-defining region of interest, the software encircles the tumor area with defined margins of 500 micrometers and 1,500 micrometers respectively. Cells were detected by hematoxylin intensity and classified as positive for the respective antibody based on their brown DAB staining intensity. Negatively and positively stained cells were counted fully automatically in 4 regions of interest: the tumor tissue, the directly tumor-adjacent invasive margin at 500 micrometers distance from the tumor margin, the tumor vicinity at 1,500 micrometer distance from the tumor margin, and the surrounding tissue beyond that border. Average cell density was measured across all those 4 regions of interest and was used for statistical analysis. These are our main findings. We first analyzed the overall density of the inflammatory infiltrate in tissue from immunocompetent and organ transplant recipients as the cumulative number of CD4 positive leukocytes, CD8 positive T cells, mature CD20 positive B lymphocytes, and CD68 positive macrophages and mononuclear histiocytes. We found that long-term immunosuppression results in a reduction in the density of immune cell infiltrates in the squamous cell carcinoma arising in organ transplant recipients, but also at all other regions of interest at the peritumoral sites. Immune cell infiltrates at the directly adjacent invasive margins were in both groups less dense than intratumoral infiltrates. Indeed, we found the lowest density of infiltrating immune cells in squamous cell carcinomas arising in organ transplant recipients. And we found the most significant reduction of the immune cell density at the invasive margin of squamous cell carcinomas of organ transplant recipients compared to immunocompetent patients. We further evaluated alterations in the phenotypic composition of the adaptive and innate immune cell subsets in squamous cell carcinoma of organ transplant recipients and immunocompetent patients, as well as their spatial distribution at the defined regions of interest surrounding the tumor. As this is a very busy slide, I would like to summarize our main findings for you. The vast majority of tumor-infiltrating immune cells were CD4 positive, which consistently outnumbered CD8 positive immune cells in both organ transplant recipients and immunocompetent patients. CD4 positive infiltrates at the directly invasive margin and at the directly adjacent tumor vicinity were significantly reduced in tissue from organ transplant recipients compared to immunocompetent patients. The lowest numbers of infiltrating CD8 positive T cells are detected within the intratumoral tissue from organ transplant recipients. CD8 positive infiltrates were significantly reduced also at all regions of interest in organ transplant recipient tissue compared to immunocompetent patients. CD20 positive B cell infiltrates also showed generally significantly reduced densities at all regions of interest in specimens derived from organ transplant recipients compared to immunocompetent patients. And in contrast to the detected differences in adaptive immune cell substance, no significant difference in the density and distribution of CD68 positive mononuclear histiocytes and monocytes was detected between the two groups. We do acknowledge limitations in the discriminatory power of lineage markers used in our analysis, as CD4, a co-receptor for the T cell receptor on T helper cells, is also expressed at lower levels by human dendritic cells and monocytes. Also, not only polarized immunoreactive but also immunosuppressive subsets may be present in the distinct populations of infiltrating leukocytes that we analyzed, such as regulatory T cells or regulatory B cells, and a functional analysis of the tumor-infiltrating leukocyte populations was not part of our study. In conclusion, our study confirms by highly objective methodologies that long-term immunosuppression results in an overall reduced density of intra- and peritumoral immune cell infiltrates of squamous cell carcinoma. Also, the local microenvironment surrounding squamous cell carcinoma of immunosuppressed organ transplant recipients obviously harbors distinct changes compared to respective tissue sites in immunocompetent patients. Our results suggest that the overall reduced density of infiltrating adaptive immune cells, particularly reduced CD4 positive infiltrates at the invasive margins of squamous cell carcinoma and reduced CD8 positive T cell infiltrates, may contribute to the aggressiveness of squamous cell carcinoma arising in organ transplant recipients. Also, the cutaneous CD20 positive B cell compartment is significantly affected by systemic immunosuppression, and this deserves further study. Why is the study relevant for dermatologists and patients? Our study provides an extended insight into the skin's normal functions and the impact of immunosuppression by highly objective and comprehensive analysis of phenotype and specific microanatomical distribution of infiltrating immune cell subsets in squamous cell carcinoma and surrounding skin, together with the integration of associated histopathological and clinical parameters. And we would like to propose that squamous cell carcinoma arising in organ transplant recipients may serve as a model cancer in which to study defective immune cell homing and function that may underlie deprived immune responses to malignancies.